So after Phycomycetes and Ascomycetes, now we are doing the second higher fungi, that is Basidiomycetes. Basidiomycetes are also called as the club fungi. Why are they called as the club fungi? You have played cards, right? There are 4 types of cards in the cards deck. One is the hearts, one is the spades, one is the diamond, and this shape is known as the club shape. And since this resembles a mushroom, that's why these fungi are called as the club fungi. Who are included in this class? The mushrooms that you eat, the common button mushrooms and the oyster mushrooms, they belong to Basidiomycetes. In the woods and all, you must have seen some fungi, bracket-like fungi growing on the stumps of trees. They belong to Basidiomycetes. Have you heard of toadstools? The red fungi with white spots which are used as umbrellas by frogs. They are toadstools. Scientific name Amanita. They are poisonous and they belong to this class. So let us write down about them. Habit. Most of them are saprophytic. They grow in soil, on stumps of trees, and they bring about decomposition. They are known to be the best decomposers of plant material like cellulose and lignin, which is a part of the plant cell wall. Even bacteria cannot give fungi so much competition in decomposing. Bacteria can decompose most of the things, but things like lignin and all in the plant cell wall, which is a very thick substance, is not easily decomposed by bacteria, but the fungi are known to decompose cellulose plus lignin very, very efficiently. So efficient that they can rot a living tree also. Best decomposers of cellulose and lignin. There is one example of a species given called Ganoderma, rots living trees. Most of the forms are saprophytic, few forms are parasitic, cause diseases like rusts and smuts. Cell wall will be made up of chitin, they are higher fungi. Mycelium will be septate or aseptate. Septate mycelium. One special thing about their septum. Okay, look at this septum that I have drawn for Basidiomycete versus the normal septa for the other classes. The normal septum is a normal septum, but in case of the Basidiomycetes, you can see that the septum has such kind of outgrowths, kind of barrel-shaped outgrowths are seen. This is a special septum. This is called as the dolipore septum. This is some Ames related information. Dolipore septum is found in the hyphae of the Basidiomycetes. Why? What is the function of this dolipore septum is, behind these outgrowths in these spaces here, they will store certain chemicals which will help to seal this opening if this hyphae gets broken. Okay, if the hyphae gets broken, then they will plug this opening and prevent the cytoplasm from oozing outside from the strand. Please note that such kind of facility will not be seen for these normal septa. They will not be able to plug in case the hyphae breaks. So all the cytoplasm will ooze out and the fungus will die. So this is a modern feature. Next, asexual spores. Asexual spores are said to be absent in Basidiomycetes. It does reproduce asexually also, but not through asexual spores. It will instead resort to vegetative reproduction. Asexual reproduction will be vegetative through fragmentation. Fragmentation is present in all the different classes, including this one. Next, sexual spores will be basidiospores, which will also be haploid spores. We will see during their life cycle how they are formed, just like how we saw for Ascomycetes. Examples: the common mushroom that you eat. There are 2 types of mushrooms, button mushroom and oyster mushroom. Button mushroom looks like this one and oyster mushroom is this one, the oyster shaped. The button mushroom species is Agaricus bisporus and oyster mushrooms species is Agaricus campestris. So these 2 species of edible mushrooms belong to this class. Next, Ganoderma, toadstools. Scientific name is Amanita. These are poisonous. Then there is one that is called Puccinia graminis. This causes the disease black rust of wheat. This is also called as the rust fungi since it causes rusts. Then there is another one called Ustilago. The disease that it causes is smuts. This one causes rusts and this one causes smuts, both of which are parasitic fungi. Next, we understand Basidiomycetes by studying the life cycle of Agaricus bisporus. So let me start the life cycle from here. As you can see, hyphae of two compatible mating types are next to each other, denoted as plus and minus. Sex organs are not seen in this one. In the previous three classes that we learned, sex organs were present. This class does not have sex organs. Plasmogamy occurs by fusion of hyphae only. So plasmogamy is happening now. Sex organs are absent in this class. Plasmogamy occurs by fusion of hyphae only. This type of plasmogamy is known as somatogamy, wherein somato means body. Isn't it body fusing? Somatogamy. After plasmogamy, what happens? So tell me, what are your observations? Once the plasmogamy occurs, the hyphae that are going to extend, they are no longer monokaryotic, they have become dikaryotic. So they are going to keep on extending, growing. They will grow as their dikaryotic hyphae. Higher fungi, plasmogamy is not immediately followed by karyogamy. Let's mark these as dikaryotic hyphae, whereas these will be monokaryotic hyphae. Mono means one. Di means they will grow. They will grow. They will grow. They will grow. Then they will grow more. They will grow some more. And then they will grow some more. And then there will be clumping of this dikaryotic hyphae. Clumping. So these hyphae are going to clump together, and they will result in the formation of baby mushroom. Okay? This is a baby mushroom, and this will grow further to form. So this is what you get after more growth and more clumping of this fungi. This umbrella-like structure is called as the pileus. Stem is called the stipe, and underneath this umbrella there will be many such kind of structures. These are called the gills. They are about 200 to 300 per mushroom. This entire structure has been made by the clumping of those dikaryotic hyphae. Karyogamy has not occurred yet. These are higher fungi. So now let us take a small portion of this gill. I'm taking a portion here. I'm keeping here like this. Inside the gill there will be the dikaryotic hyphae scattered. Then on the edges there will be slightly more clumping, and towards the edges you will see such kind of structures. These are the terminal portions of the hyphae. So let's analyze this one, immature one. This is the terminal hypha. Terminal part of hypha. Is there a dikaryotic situation? Yes, dikaryotic situation is still present. Karyogamy has still not occurred. What will happen now is, in this terminal cell, karyogamy. Has karyogamy happened? This is 2n nucleus. These two nuclei were NN. Each of these nuclei were NN. And after karyogamy, 2n nucleus has formed. After plasmogamy, finally karyogamy has happened. Next step will be meiosis. So 4 NN nuclei have been formed. So the 3 steps of sexual reproduction are done right here. What will happen now is, look, these 4 nuclei have emerged outside from this terminal cell and they have rested on this structure. These 4 nuclei have now become basidiospores, which will be haploid sexual spores, and the structure has now become basidium. When the gills split, all these spores are going to be thrown out. These basidiospores will be thrown out in air to start a new life cycle. This is basidium, which is inside this fruiting body. This is its fruiting body. What is this fruiting body going to be called as? Basidiocarp. Carp means fruit. What we eat are the fruiting bodies. Compare the basidiospores with the ascospores. Basidiospores, compare them with ascospores. These were endogenous. These are exogenous. Ascospores were always 8 and basidiospores are always 4. Why 4 and not 8? Because in ascomycetes, the nuclei that were formed after meiosis had undergone one mitosis to create 8. That's why we had got 8 per ascus. But over here, these 4 nuclei are not undergoing mitosis. They directly form the basidiospores. That's why only 4 per basidium. 4 per basidium, and ascospores are 8 per ascus. Good. So these are Basidiomycetes, the higher fungi, more advanced than Ascomycetes in having dolipore septum. They are higher fungi. After plasmogamy, the karyogamy is very delayed. A prolonged dikaryophase will be seen in them. Prolonged dikaryophase seen. Finally, there is karyogamy and meiosis, and basidiospores will be formed in the basidium in this manner. And once the fungus just opens out like this, spores will be dispersed in the air, and each spore can form a new fungal colony. Clear?
महादेव के नाम पे सब्सक्राइब कर दे।